GIW Singapore. And good morning from Alabama. Today I'd like to talk about PharmAlchemy, which is a comprehensive knowledge base for mining gene-drug disease relationships. This work was conducted by the Spark Systems Pharmacology AI Research Center at the University of Alabama at Birmingham in the United States. So what exactly does the lab that I'm part of work on? I'm a first-year PhD student. My background is in biology as well as computer science. So as part of this lab at UAB, we develop translational bioinformatics as well as AI and machine learning tools and databases to advance precision medicine in cancer and other complex diseases. For instance, we work with genomic data, as well as pharmaceutical data, as well as disease data and electronic health records, integrating them into a multi-omics application. In addition, we work on systems biology and network modeling. How does a drug affect the target that leads to some kind of phenotype that contributes to disease? Furthermore, we also do quantitative and qualitative analysis of gene expression as well as clinical phenotypes using various statistical tools that we've developed in-house. In addition, model simulations are some of the things that we are working on. How can we create systems that allow us to simulate drug response using artificial intelligence? And overall, the goal of all these efforts is to contribute towards a low-cost method for individual patients to achieve personalized precision therapeutics, and in this case, oncology, precision oncology. So just focusing in on data-driven medicine, how has it been applied to this field of oncology? Well, currently, multimodal data sets are crucial in order to contribute towards the analysis, recommendation of drugs, as well as disease monitoring in response to chemotherapy, disease states, patient diagnoses, as well as the recommendation and tailoring of personalized therapeutics. Some of the various forms of data sets that have been used and integrated towards multimodality include patient history, EHR, tumor biopsies, and microarray expression, as well as the tumor microenvironment, patient-to-patient similarities, as well as comorbidities, as well as different forms of drug response, including drug resistance. So here we have a problem and a motivation for integrating large-scale databases. It appears that most of the currently existing data sets are not sufficient, or either not sufficient or not well integrated for answering scientific queries involving drug repurposing or personalized medicine. Yes, there are data sets such as DisGeneNet, CTD, DrugBank, PharmacoDB, and DMAP that have noted several tens of thousands of genes, diseases, variants, as well as interactions between drugs, genes, and diseases. That being said, they have not been well integrated into an all-in-one application that allows for the answering of scientific queries regarding personalized medicine and precision oncology. So here we present PharmAlchemy, which is a vision for unifying representations of entities and relationships between genes, drugs, and diseases. So traditionally, we have seen that drugs have some kind of an effector relationship with genes, whether that's through pharmacology or some other mode of action, activating or blocking some kind of receptor or modifying some kind of enzyme. Genes have been associated with diseases, and the presence of upregulated or downregulated gene expression has been shown to correlate with different phenotypes. In addition, drugs and diseases have the impact of drugs on diseases is kind of like the first step towards affecting some kind of downstream phenotype. That being said, there are many possibilities when it comes to pairwise combinations between drugs, genes, and diseases. Not only drugs, genes, and diseases, but also drug combinations, gene sets, and disease cohorts, as you can see in this figure. In order to develop the first iteration of PharmAlchemy, we relied on the integration of various data sources of drugs, genes, and diseases, as well as drug targets, as well as drug-to-drug interactions, side effects, and other gene-drug disease interactions. We performed several months of data collection, data cleaning, standardization, missing value imputation, removing duplicates, standardizing primary foreign keys, and making sure that the data was using the correct identifier for various entities in the databases. And overall, we uploaded everything into Oracle, Application Express, and that's what we used for this application. We designed an innovative database schema that allowed for the correct identifiers so that we can have not only disease, drug, and gene information, but also pairwise combinations of each of the different tables from aggregating from all the different data sources that we had collected over the past few months. We ended up with 56 million drugs in our database, as well as 81,000 genes and 721,000 non-drug therapeutics, covering about 17,000 individual diseases, as well as various pairwise combinations of drugs, diseases, and genes, including about 2.5 million multi-species gene-to-drug interactions, and 12.6 million gene-to-gene interactions, specifically from the string database and other data sets, as well as 18.3 million drug-to-disease interactions. Are specifically treating cancer. So we have about 45,000 drugs or drug sets that pertain to pharmaceutical treatments for cancer, and about 9,000 other, 8,700 procedures, and various other treatments that allow us to have fantastic coverage for cancer-related illnesses. So the PharmAlchemy application plans to make some of these questions easier. These are some of the scientific queries that we have addressed so far in our application. So if a user were to pull up PharmAlchemy, and they would be able to ask a few of these questions, like, PharmAlchemy, what are some of the known cancer subtypes associated with certain patient gene expression? PharmAlchemy, let's say my patient has a certain gene expression for their biopsy. What are some of the candidate drugs that could reverse some of the changes of those cancer subtypes? If my patient is also taking some of these reverser compounds recommended by PharmAlchemy, what are some of the other concurrent prescriptions taken by other patients in the database who are also taking these reversers? PharmAlchemy, what recommended drugs have the best disease coverage for a patient's set of diagnoses? Let's say a patient has diabetes, kidney disease, breast cancer, and how could we recommend drugs that have the best coverage for the entire set of diagnoses according to the indications in the database? PharmAlchemy, what are some of the drug candidates with similar molecular fingerprints and gene targets, along with the mildest side effects for this patient? So we aim to implement an algorithm, and we have implemented some of it thus far, in order to obtain reversal drugs. We would define reversal drugs as drugs that can upregulate the downregulated gene expression and downregulate the upregulated gene expression of some kind of a tumor biopsy. That would be an example of the use case for this algorithm. And so the input would be a gene set of expression values from a tissue sample, as well as clinical diagnoses of the patient. And then it would perform a reverser drug search to find ideal drug candidates based on these considerations, including disease coverage, molecular fingerprints, as described previously by the gentleman before me. Molecular fingerprints are bit vectors of ones and zeros that correspond to the functional groups and structure of various pharmaceutical compounds. Gene targets are also considered in the drug search, as well as side effect profiles and clinical trial status. The output of this algorithm is a list of drug candidates that would be recommended individually for the patient based on their patient-level tumor biopsy, gene expression, as well as clinical diagnoses. So let's look at a case study performed using samples from triple-negative breast cancer tissue. So after performing differential gene expression analysis, we identified that there were about 10 genes that were significantly upregulated in the triple-negative samples, as well as about 10 genes that were downregulated in the triple-negative breast cancer samples. And you can see here is a volcano plot that shows that there were many genes that were significant at the FDR less than 1% significance. So I would like to demo PharmAlchemy, and PharmAlchemy here is it's accepting the upregulated genes and downregulated genes from the differential gene expression analysis. When I click on Generate Reversers, it will bring up this screen that will show the various reversers that have been queried using an API, and that API has pulled these various reversers that will reverse that downregulated and upregulated gene expression. If I continue, I'll be able to obtain a list of all the drugs in the database as well as their indications. So as you can see, all the aspirins are grouped together by their various trade names, and all the other drugs, they are grouped together according to their indications. Interestingly, we are able to see that the patients who are taking those reversers are also taking other drugs, and so we would see the concurrent drugs taken by similar patients who are taking those reversor compounds. So this is a list of the concurrent drugs taken by patients who are also taking those compounds recommended by the API. And then if we do a search for cancer-related compounds, we see that there are various compounds taken by similar patients, such as colon cancer drugs as well as thyroid cancer drugs. The key feature of PharmAlchemy is the comparison of molecular fingerprints with DrugBank or Supernatural, looking at databases of 40,000 or 80,000 drugs in this prototype, PharmAlchemy 1.0. And so if we were just to focus on one of the reversers, let's say, for instance, mibifridil, which is a known blood pressure medication, what would happen if we looked in the data set and we looked for compounds that were molecularly similar to mibifridil? Well, what happened is we would be able to get a list of similar compounds ranked by their similarity score, as well as the ability to check for their side effects. But it gets more interesting. Here we see that 4 of these compounds are known anti-cancer compounds. We see that buparlisib, which is molecularly similar to mibifridil, is a known anti-cancer PI3K inhibitor, as validated by manual literature search. We also see that there's an experimental compound that targets DNA synthesis. Ilarasertib is a known aurora kinase inhibitor, and we see that there's also a known anti-breast cancer protein kinase inhibitor that we have identified using PharmAlchemy, using the molecular fingerprint method. So to summarize this case study, PharmAlchemy is an exciting, new, comprehensive knowledge base for looking and examining gene-drug disease interactions. We've demonstrated that one of the many applications that has implemented a reverser drug search algorithm, and we have implemented it as seen in this demo. There is a limitation, and for future work, we have to continue working on the user interface, as well as working on database coverage, making sure that we implement additional features, especially disease coverage and integrating with electronic health records. And we hope to do that. We hope to do that really quickly, as soon as possible. Thank you.
Thank you. Now we take questions from the audience. Any question? Okay, please.
Thank you. Thank you for the nice work. I just want to ask, I think you didn't, maybe I missed it, like what's the algorithm to sort of prioritize the different drugs? So what makes one drug ranked above the others? So maybe you can describe a little bit about that.
Yeah, so this is a prototype application, and we looked at the Jakarta index between molecular fingerprints. Yeah. So we use the Jakarta, which is intersection over union for the 0-1 bit vectors.
So, but you considered other aspects besides just comparing the molecular fingerprints, right? So how do you integrate those different aspects to sort of prioritize these drugs?
Yeah, so other aspects would include looking at gene targets, as well as the side effect rankings. As you can see, there's a, not here, but on another screen, there would have been a ranking of severity of side effect. But in order to answer your question, we did not implement this, but we would have looked at most common intersection of gene targets and lowest ranking of side effects from 1 to 3. Yeah.
Okay, thank you.
Thank you.
The last question.
Surinder Gichi, I'm a program director at National Science Foundation. So the question is this available or not yet?
It is.
I did a search and I ended up on an Italian website selling essential oils.
It is, we have a private link, but it's not ready for deployment, but we've been testing it. We have a URL, but we're still working on it right now. Yeah, it's been my first semester's work has been working on this. So I feel like, to answer your question, we do have a link, but we're not ready to release it.
Understood. And what is the plan? Do you plan to make this available, for academic purposes? Are going to have an API? Are going to have a web tool?
I think we do plan on releasing this as a web tool. I know that the lab I'm in has an extensive record of releasing previous tools. And so I would think that we would want to release this to the scientific community.